Labib, thank you. Good morning, ladies and gentlemen. Thank you for being here. So my name is David Lefebvre. I'm from the Scienzano Institute in Belgium. I will tell you something about foot-and-mouth disease in Nigeria. What I will tell you is actually a summary of 4 different abstracts in the abstract book. 3 of these abstracts originate from OIE, twinning collaboration between my own institute, Scienzano, and the National Veterinary Research Institute in Nigeria. And the other abstract originates from a collaboration between the National Veterinary Research Institute in Nigeria and the Plum Island Animal Disease Center in the United States of America. So Nigeria is this very big country in the west of Africa. It has the same size as France plus Germany, but the number of inhabitants is much higher than of France plus Germany. So it's a very densely populated country. It's the largest African economy, but it's highly dependent on the exportation of oil and gas. The agriculture in Nigeria is still very much subsistence oriented, and the number of livestock which is kept is relatively low compared to the number of people living in the country. So there is a demand for animal products which largely exceeds the domestic production in Nigeria. Consequently, there are significant movements of livestock across the borders and also inside the country due to the importation of livestock, but also due to the more traditional methods of keeping livestock. There is no systematic surveillance for FMD in Nigeria. Notifications of FMD are only made very sporadically. There's no control program, and vaccines are not locally available. On the previous open session of the EuFMD, one of my colleagues from Scienzano and also colleagues from National Veterinary Research Institute in Nigeria, they presented previous results. And basically in the period of 2012 to 2015, we were able to detect 4 different serotypes and in total 5 different topotypes in 4 provinces of northern Nigeria during this period. And these sero and topotypes were O West Africa, O East Africa 3, A Africa G4, SAT1 topotype 10, and SAT2 topotype 7. Now, the epidemiology is, as in any endemic country, it's quite complex and highly dynamic. So for an example, in the plateau states, so Nigeria is a federal country with different states. The country itself, as I already mentioned, is very big, and the size of a state is somewhat comparable to a smaller European country like Switzerland or Belgium or the Netherlands. So the plateau state where the research institute from our colleagues is located right here. And if we look in the period from 2013 to 2015, we see that each year there is one other serotype that is the most prevalent. So this was SAT2 7 in 2013, O East Africa 3 in 2014, A Africa G4 in 2015, and then it was SAT1 topotype 10 from the end of 2015. Now, particularly, the outbreak of SAT1 was very interesting because the previous outbreaks with SAT1 in Nigeria, they already date from the topotype 5 outbreak in 1967 and the topotype 6 outbreak in the period from 1979 to 1981. It was also a very special strain that we could isolate because the genetic characterization showed us that the SAT1 isolates from 2015 it's not cluster with any other previously documented strains, so they were characterized as a new topotype 10. Now I will show you what we have been doing in the period of 2016 to 2018. So first of all, we performed studies on epithelial samples from suspected FMD-infected cattle, which were collected in northern Nigeria, but also southwest Nigeria. So 45 samples from 2016 from 3 different states, and then 3 archived samples from 2014. were analyzed in Plum Island. Now, how does it work? Actually, the people from Nigeria, they collect the samples in the field. They do a primary characterization with them. So they collect samples, and then they select the best samples to send them to another lab to be able to perform the genetic characterization. Then samples from 2017 from 7 different states, they were sent to Cienzano, and as well 33 archived samples from 2012 and 2014 from the Cachia Grazing Reserve. They were also sent to Cienzano. So I already said that livestock in Nigeria is mostly kept in very traditional ways. This involves a lot of long distance movements with seasonal cycles of the cattle. But in an attempt to force the people to be more sedentary, the government, since quite some time, has also installed so-called grazing reserves, in which are predominantly reserved for grazing animals. So this comes from one of these grazing reserves. Now, besides that, we also analyze serum samples from different serosurveys, serosurvey in sheep and goats from 2014, serosurvey in wildlife from the period 2009 to 2014. The animals involved were waterbuck, wildebeest, and African eland. Then also a larger serosurvey from the Cachia Grazing Reserve with more than 1,000 samples in cattle and more than 100 samples in sheep, but also serum samples from 2 FMD outbreaks in the Cachia Grazing Reserve from 2012 and 2014. So, which are the results then from these epithelial samples? So, the samples from 2014-2016, there it could be found again the presence of O West Africa, O East Africa 3, A Africa G4, SAT110 and SAT27, which are exactly the same sero and topotypes that were previously documented. In the more recent samples from 2017, we could only observe O East Africa 3, A Africa G4, and then SAT27. But actually, the most interesting finding was that in one state, in a period of only 3 months of time, we could identify outbreaks with 3 different serotypes, so 3 different serotypes in a single state in only 3 months of time, and in another state, we had in 2 months of time also outbreaks with 2 completely different serotypes. While on the other hand, we could also see that a certain sero/topotype could be isolated at exactly the same time in 3 completely remote states in the country. And then the samples from the Cachia Grazing Reserve, they showed an outbreak of SAT27 in 2012, O East Africa 3 in 2014. Now the Cachia Grazing Reserve is located in a state next to the state of the Veterinary Research Institute, and these data make a lot of sense because in the plateau state, the most dominant serotype in sero/topotype in 2013 was on the SAT27, and in 2014, so again in this neighboring state, they also noticed East Africa 3. Then the results from the serum samples. So what did we learn from the serosurveys? So in the serosurveys in general, we noted a lower prevalence of antibodies against foot and mouth disease virus. These were antibodies against NSP as well as antibodies against B. In general, in sheep and goats compared to cattle and wildlife. This is also a consistent finding that we see in other studies. And also most sheep and goats that were investigated were seropositive for just 1 single serotype, while significantly more cattle and wildlife are positive for 2 or more serotypes. That's what you also see in other studies. Then when we look into the serological samples, until 2014, there is clear serological evidence for circulation of O, A, and SAT2 in the country. So I already mentioned that the SAT1, which was identified in 2015 with humic new introduction, because it wasn't documented SAT1 outbreaks since 1981. And also, so in the serological data, you don't see any evidence of the presence of SAT1 until 2014. Then serum samples from the FMD outbreaks in the Kakia grazing reserve. So until 2012, there is serological evidence for circulation of O and A in the KGR. And then in 2012, so they had the outbreak of the SAT2 topotype 7. And indeed, if you look then at the samples from 2012 onwards, you also see serological profile being positive for this SAT2. So, what can we conclude, and what would be nice to discuss a bit about, is that in the samples from 2017, we couldn't isolate O West Africa, and we couldn't isolate SAT1 group topotype 10 either. So the question is, have these strains been outcompeted by the three others, OEA3, A Africa G4, and SAT27, or are these topotypes still circulating in the country, but do our colleagues from the NVRI don't have access to samples from states in which these topotypes are circulating? You probably know that Nigeria, from a political point of view, is not the most stable country the most recent years. So it could be that O West Africa and SAT110 are still in Nigeria present, but that we just don't see it because they are on places that are not sampled. So, as I mentioned, until at least 2014, there's no evidence of circulation from SAT110. So we can still assume that SAT1 was indeed a new introduction into Nigeria in 2015. But unfortunately, we don't have any data available from neighboring countries, particularly Niger and Chad, which act as an important access road for importation of live animals that mostly come from more eastern parts of Africa. And also very interesting is the lower prevalence of FMD in small ruminants. This is also observed at a household level. How does this come? Huh? Is this because small ruminants are or maybe less susceptible to infection with FMD virus than cattle? Or is it because they just mount lower antibody responses, which are thus less detectable? Or maybe it's because small ruminants have a larger number of offspring, and consequently, the age structure of the herd is different. The herd is relatively younger, and because of that, it's maybe that there is less seroprevalence. So if you have the answer to one or more of these questions, please tell me. So I want to acknowledge the OIE for supporting the laboratory training program between Sano and NVRI. Also the EUFMD that supported these studies through the FASTEP project. I also want to thank all the collaborators to the study, so the people from the NVRI in Nigeria, their colleagues from the university, Ahmadu Bello in Nigeria, my colleagues from Sano, and then the colleagues from the Plum Island Animal Disease Center in the United States. And so I thank you for your attention. I thank you also for being here, of course. And if you have any questions or remarks or answers to my questions, just tell me, please. Thank you.